Sue has a question around analytics, and I've got a couple questions there too. And I know that this response is probably varied, so I'll ask you to maybe give us some context to your answer. But she'd like to know which analytics are most important to driving process intensification. As I said, I'm sure that there are different responses depending on the operation that you're addressing. But if you can give us some context and share your response there, Irina, maybe I can ask you that one first.
There are two different ways that the bioreactor can be operated as a perfusion. A lot of those that have experience for the last 3 or so decades are used to look at a perfusion bioreactor that lasts several weeks, and they will expect the titer to be constant. And that actually makes it requiring the titer measurement real time less important. But for those, they are actually running a very high cell density bioreactor where the titer changes, and we want to capture the product from the beginning and we don't wait for a steady state. We actually require to know how that titer change can impact the downstream capture. So having analytics or a PAT or something that goes, for example, ramen has been evaluated, that can tell us real time, and hopefully through digitalization can inform the downstream system real time, what's that titer when the column is being loaded, even during that load phase. That's super critical. So you utilize the resin accordingly to your capacity. So that's an important one. Another one that might come to the list to me is very close to the importance of process control is how can we detect microorganisms if there is any growth sooner than the traditional method that we have to wait for days. Mm-hmm. Because from my experience, a drop leaving the bioreactor takes about 24 hours in average to reach the drug substance back. If it takes over a day for you to detect microorganisms growth, which is the same as contamination, in any part of that process, you're done. Your process is all in jeopardy. You have no time to react. You cannot divert material to waste without having to clean the entire process. You better just start over. And as we heard before, the management of volume, the sizes of vessels, and returning to train, that's pretty complicated to assess in a facility. So those are, and I'll stop here, but those will be my two main points, at least to start off, and then we can talk about others.
Thank you, Irina. Sanjay, what about you? Any other thoughts on which analytics are most important?
Yeah, I think the product quality aspects for the step, say if you're using a continuous chromatography, so what you get from the normal chromatography to continuous chromatography will be what kind of a product quality attributes you get from each unit operation, whether impurity clearance and product quality, yield, and stability of your intermediate. I think that would be important for me to consider. Mm-hmm. That's one of the aspects for, yeah.
Yeah. Sanjay, in these examples, what give us a sense of what systems or applications you're using to monitor, record, report on these analytical points.
We use multiple analytical techniques, something like aggregation, non-reduce and reduce bioanalyzer or CGE is another one, and impurity clearance, HCP, DNA, and product and process-related specific impurities. That's some of the tools we use to measure the process, process performance. And we would like comparability between a continuous or the intensified manufacturing or continuous manufacturing to your traditional kind of a manufacturing. That you have to show the similarity or comparability between the process you have between both modalities.
Sanjay, those are all measured offline, right? Those are the So we leverage those that sort of platform analytical toolbox, if you will, the same way that we do for batch to continuous during process development, and it's through the control of the parameters of the operational design of the process that we guarantee that the product is safe and high quality. Of course, then for release, we would have to monitor that, sampling every day in particular points of the process, during process development, during the scalability, both for pilot and then eventually to clinical stage. But there's new technologies that are very promising, from Raman to IR, that they are looking into aggregate levels, fragment levels, concentration of excipients, concentration of our protein throughout just a flow cell, wherever you want to place it. Of course, it's all questionable until you try it. And it's also, there's, I'm sure, some limitations on which ranges can it actually pick it up. So we have several initiatives in collaboration with our analytical teams to look into these novelties, to get some of these vendors to come over and show us how it works. And of course, tell us our wish list. It's a two-way street, right? They promise the technology is capable of a certain type of detection. They show us their preliminary data on their own matrices and samples. We test with ours, and then we say, by the way, we want to implement it in a particular setup, and we want to integrate digitally these results to now inform what we want to do next. And those last, the latter part of the strategy is not implemented yet, because I believe we're all growing together. Does that make sense? It's like it's almost like this community giving the visibility and, of course, the important drivers for these particular processes. He's growing together, and this is great to be part of, I must say.